Actually a very big topic, sir. We are done with very big topic, that is about the proteins topic. Protein topic has been completed. Now let's move on to the next topic, that is enzymes topic. Enzymes is another favorite topic of the examiner, sir, MC examiner. Many questions will come from enzymes topic also. Let's see what they'll be asking. Now first of all, what is asked in the enzymes topic is about the Km value. Now what is this Km value? First of all, look at this one. In this graph, we are taking a relation between velocity of the reaction, okay, capital V here, velocity of the reaction, and then we are taking a graph between the concentration of substrate. Concentration of the substrate. We are taking a relation between them. And we all know velocity of a reaction is directly proportional to the concentration of substrate. So more the substrate, more will be the velocity. More the substrate, more will be the velocity, guys. Is it okay? And this is actually given in Michaelis-Menten graph. It is given by Michaelis-Menten graphs. Let us learn very quickly, guys. Every topic, whatever I'm telling you, focus only on those points. At least revise this and go. Okay. And many of you were asking me, yes sir, revise these points only and go to the exam, sir. More than enough for your exam, at least at this very high point, high time, sir. So what is this Michaelis-Menten graph? Michaelis-Menten graph will give you the relation between the velocity of the reaction and substrate concentration. And we know directly proportional. So that is the reason why we are getting a parabolic graph. Now in this parabolic graph, where are you getting the maximum velocity? So maximum velocity is somewhere here. That is the Vmax. Okay. Now what about the Km value, sir? Km का मतलब क्या होता है? Km is nothing but substrate concentration only, but when? At half the maximum velocity. Sir, इसका मतलब क्या है? What is the meaning of this one here? First of all, Vmax is here. What you do is you take like half the Vmax by 2. Half the maximum velocity. At half the maximum velocity, how much ever will be the concentration of substrate? This is actually referred to as your Km. I'm not going into much detail. अभी आपको जो चाहिए वो मैं आपको बता रहा हूँ. Whatever is required right now, I'm telling you that one, sir. What is Km? Km is nothing but substrate concentration at half the maximum velocity. Substrate concentration, guys, at half the maximum velocity. Some of our friend there, Dr. Rajat and everyone, they are suggesting you to refresh there, guys. So try to refresh the video at your end. At my end, everything is perfect. At my end, everything is perfect here. Please try to refresh the video at your end there, guys. Okay. चलिए आगे बढ़ते हैं. तो I repeat for the last and final time here. Michaelis-Menten graph will actually give you the relation between the velocity of the reaction and substrate concentration. It is directly proportional, and half the velocity, half the maximum velocity, that's nothing but the Km value, sir. The Km value is defined as at half the maximum velocity, whatever is the substrate concentration there. चलिए आगे बढ़ते हैं, sir. Let us go ahead right now. Now, sir, what is the use? Now some of you might be getting a question in your mind. Sir, इसका use क्या है? Why should I know about the Km value and the Vmax value and all those things? Welcome here. I will tell you the question which is going to come on 18th June. That is about the inhibition, enzyme inhibition, reversible and irreversible enzyme inhibition. There will be two types of enzyme inhibition. Pakka question, sir. Pakka. Definite question in your 18th June. Pakka question, sir. Okay. Now, reversible inhibition will be again of two types. One will be the competitive reversible inhibition here. Another one will be the non-competitive inhibition here, sir. One is competitive inhibition here. Another one will be the non-competitive inhibition here. अब यार ये competitive और non-competitive में क्या होता है? What is going to happen in this one? You have to remember the Km value and the Vmax value. Okay. In competitive inhibition, remember the Vmax value will remain the same, whereas the Km value will actually increase. Are you learning this one here, sir? So Vmax value will be remaining the same. Okay. Yes, G. Come on. Competitive inhibition में क्या होता है? What is going to happen in competitive inhibition? Come on, my dear friends. Vmax value is going to remain the same, whereas the Km value will actually increase in competitive inhibition. Competitive में ये increase होता है. Exactly opposite of that in non-competitive inhibition. In non-competitive inhibition, Km value will be the same, and the Vmax value will actually decrease. Vmax value will decrease here. Now, in one of the class, I remember some student told me, sir, what is this Vmax same, Km value increasing and all those things? Okay. What is this Vmax same, Km value increasing and all? Someone asked me here. Actually, there is a big science behind that one. ये पूरा ग्राफ है, the same graph here between the velocity and the substrate concentration we are taking. This black colored line is actually, yes, with the inhibitor here. The red colored one is without the inhibitor there. So we are trying to check with the inhibitor and without the inhibitor. So this graph, the very first one is competitive one. Sir, how can you tell in this competitive one? You can see both of them, the graph number 1 and the graph number 2, with inhibitor, without the inhibitor, both of them are having the Vmax value same, sir. So same Vmax, what is happening, sir? Competitive inhibition. Whereas the Km value is actually increasing there. Understanding? तो इसीलिए मैं आपको कह रहा हूँ that is the reason why I'm telling you here, guys. You don't have to worry about that all that big graphs and everything for your MCI exam right now. Okay, it's very high time. Rapid revision. इतने याद रखो, काफी हो जाएगा. So in competitive inhibition, Vmax same, Km increases. Non-competitive inhibition, Km same, and the Vmax will actually decrease. हाँ, look at that. Dr. Sultan Ahmed there, he's telling competitive में काम ज़्यादा होता है. Competition में क्या होता है सर? Competition में काम ज़्यादा होता है. Km value actually increases. Competition में क्या होता है? काम ज़्यादा होता है. ठीक है? So therefore, what is true about the non-competitive? Non-competitive में क्या होगा? What is going to happen in non-competitive? The Km value will remain the same and the Vmax value will actually decrease. So therefore, lower Vmax. Lower Vmax. No, not irreversible. Irreversible में ये लागू नहीं है. It's not applicable, Dr. Whipple, in irreversible one. This both of them come under the category of reversible only. Reversible inhibition is in turn divided to competitive and non-competitive one. So you have to just remember about this one for your MCI exam. Is it okay, Dr. Whipple? Let's go ahead. Pakka question, definite question is going to come here. Next one, sir. What happens to LDH1 and LDH2 ratio in myocardial infarction? Hello, my dear friends. Another update here. Nowadays they are asking even about the isoenzymes. Nowadays they are asking questions regarding the isoenzymes in your exam. And for isoenzyme, you have to remember one example, that is LDH, lactate dehydrogenase. How many isoenzymes do you have, sir? 5 of them. That is your LDH1, LDH2, LDH3, LDH4, and then LDH5 are there, sir. There are totally 5 different types of isoenzymes. LDH has got 5 isoenzymes. LDH1, 2, 3, 4, and 5. Now in this, what you have to remember is LDH1 and 2, these two are mainly present in your heart. LDH4 and 5, these two are mainly present in your muscle, skeletal muscle, whereas LDH3 is present in remaining all the organs there, brain, the spleen, RBC, etc, etc, etc. Is it okay? So LDH1 and LDH2 are there present in the heart. Now sir, why is this isoenzymes important nowadays? अब ये ज़्यादा important क्यों है? Why? Because my dear doctors, remember nowadays clinical questions, they are trying to always test your clinical knowledge. So for clinical knowledge, my dear friends, they are asking you regarding this one here. And I hope you have learned about myocardial infarction, MI. I hope you have learned about MI, myocardial infarction in pathology or internal medicine. And there I think you have learned about the flipping, LDH flipping, guys. Yes. So what will happen to LDH ratio in MI? In MI, LDH1 will be actually more when compared to LDH2. In the normal human beings like you and me, in healthy people, actually LDH2 will be more when compared to LDH1. But in MI, what is going to happen? There will be LDH flipping will occur. LDH flipping hota hai, and in that, what is going to happen, guys? LDH1 is actually more when compared to LDH2. Lovely. Achha, not only that, you also remember one more thing, that is your creatinine kinase. Creatinine kinase has got like 3 isoenzymes, and those 3 isoenzymes will be CKMM, another one will be the CKBB, and another one will be the CKMB, guys. नाम से ही समझ में आ रहा है सर, CKMM will be present in the skeletal muscle, whereas CKBB, this will be actually present in the brain, whereas CKMB, it is the one which will be present in your heart. And again, it has got an importance in your MI. Understanding? So for isoenzymes, I want you all to remember these 2 things, already given in the recent exam. So in enzymes topic, if you remember this, my dear friends, more than enough, sir. Okay, remember about isoenzymes and remember about the nature of enzyme. Nature of enzyme तो आपको already पता है, about the protein in nature and even the non-protein part of the enzyme. And then you also remember about the, yes, the reversible inhibition and irreversible inhibition, especially the competitive and non-competitive, guys. Okay, so I'm just right bang on. I'm telling you exactly the topics that you have to remember for your rapid revision. Okay, so if you want to remember or if you want to learn the entire biochemistry, you're always welcome to my regular classes. In the regular classes, I'll teach you the entire biochemistry. But right now I'm telling you bang on whatever is just required for 18th June exam, sir. ठीक है ना, बस ये पढ़ो आप, काफ़ी है। नेक्स्ट वन,